Yes, so before we go into this photosynthetic cell factory concept, I'd like you to think about a leaf for a moment. It's a green thing that does the photosynthesis. You know, carbon dioxide goes in, sugars come out, and oxygen comes out, you know, photosynthetic microcompartments, thylakoids, chloroplasts, all these things. So basically what we're gonna do, we're gonna put microalgae or cyanobacteria inside different bio-based and biodegradable materials that mimic the leaf, like you know it. And then we produce chemicals or a lot of different things. And like Nina said, this is a big collaboration and project between different research facilities all across Europe, as well as in Finland. And we have a lot of expertise in, for example, material science and biotechnology and synthetic biology even. So let's go through the concept first. Like you all know, non-sustainable use of natural resources is probably the biggest problem of the 21st century. And because of that, there is a big demand for green technologies. You know, you don't want your river to look like this or your sky to be gray like that. So with biotechnology and synthetic biology, with sustainable solutions, we could probably turn the environment back into this, yes. So each and every day, yeast, fungi, animal cells, bacteria, they start their shifts as cell factories, producing different chemicals to our needs. And we would like to make it even more greener. So we would use photosynthetic cells to do this production of biofuels or monomers for polymers and fine chemicals and even pharmaceutical ingredients. And there is basically no need for organic feedstock because the cells, they take carbon dioxide and then they do the products. So we don't need to put sugar inside the system. There is also no competition with food production or land use for that matter. And one thing that's very fundamental in this approach is that we don't want to do biorefinery approach in this system. So we don't want to produce the cells in a large quantity and then extract the things out from the cells. We would like the cells to be continuously producing the products that we want to get out from them. All this intricate biology in this system, it's indeed done by University of Turku in Finland, and we at VTT, we specialize in the immobilization and the solid state part of this system. So here you see, basically, usually we cultivate photosynthetic microbes in a suspension like this, and in this project we're gonna immobilize the cells, put them stationary in different matrices to produce these solid state cell factories. And why we want to do it? Well, there's a lot of benefits to this system, and I'm gonna go through them now. First one being reduced water and energy consumption. You know, the cells are stationary there, so we don't need to have a lot of volume of medium to just mix the cells around. Normally you would have some cells that are in the dark inside the vial, some are in the light, and then you would need a lot of energy to mix the cells so you get efficient production out from all of the cells inside. But like this, we only need some media to put the nutrients inside the matrix and to get the products out from there. And then there is also less need for mixing because the hydrogel is stable there. And this also leads to the second point, which is improved light utilization efficiency. So because the cells are stationary, they're always constantly in the same light that we can select regarding the needs of the algae or the cyanobacteria. You know, some need higher light, some need lower light, some need specific spectra of light, and so on. And then also, this is interesting. We have seen that there's increased cell density and cell viability inside these hydrogel films. And I think it might be because it kind of feels like it's a microbial or algae colony or bacterial biofilm that's being emulated here in this immobilized system. And also because the cells are spatially restricted, there is not that much biomass growth as in suspension, but on the other hand, I think we can direct the metabolism towards higher product formation then. But in order to get this thing working, we need good materials, we need engineered cells. It's a demanding system, you know. The photobioreactor, it doesn't operate itself, so to speak. So in Futura Leaf, we really try to make these leaf mimicking structures to really get to the bottom of this and get the thing working inside the photobioreactor. So basically what we have multi-layered structures with a porosity density gradient like this. So in the top we have denser layers of materials where we can arrange the cells in different ways. We can have different light harvesting complexes in different cell types, so that they take first the highest intensity of light, and then the scattering rest of the light goes to the longer light harvesting complex algae towards the middle of the material. And then in the bottom we have more porous materials that can convey all the ingredients to the cells, and also the products out from the cells. So how do we do it? We have different nanocellulosic materials. We have films, we have hydrogels, cryogels and aerogels, which we can produce and arrange into these structures, hopefully in the future. So maybe let's look into the matrix a bit more, because I'm from a nanocellulose team at VTT, so we're really trying to do our best to get all different kind of materials that we can implement in this system. So I don't know how familiar you are with nanocelluloses, but there is a lot of different grades of it. Now I'm going to talk about cellulose nanofibers. So they have one dimension in nanoscale, and then high aspect ratio. They're very long fibers. And they can be more coarse, like you can see that foam over there, or they can be very transparent, like hygroscopic hydrogels like this tempo pre-treated or tempo oxidized cellulose nanofibers, or TZNF. And it's really interesting material for our purposes because it has a high mechanical strength, it's transparent, tunable porosity, and it's very accessible for modifications. But this water interaction, it's also a problem. Now if you think of a paper and you put it in water, what happens? It will turn into this sludge. So we have to make this stable at aqueous conditions first. And how do we do it? We have mainly two different means to do it. If we make films, we can do chemical cross-linking. Here we've done esterification with PVA. So basically the fibers are attached with esterific bonds. But this requires completely dry environment, so it doesn't work in the hydrogel. But in the hydrogel state, we can use divalent ions to gelate the gels, and then they stay stable in aqueous conditions. Alright, then let's look into a case study. This is published data and something we've already done, and it's looking pretty nice. So we used this experimental design where we had the TCNF material I showed you before, and then we compared it to like usually conventionally used alginate hydrogel material, and then we produced ethylene, and ethylene can be used as a precursor for bioplastic formation, for example. So it's a volatile compound. It comes out from the gel, so we don't need to take the cells out from the gel. The cells will make the product and it will come out itself. So like this, we first have the starting material, we cross-link it, and then we did the production assay. And we also measured different properties from these gels, which I will show you now. Here's how they look. They're both self-standing, a bit different though. The alginate looks a bit more like, it's very dense and not very rough, whereas the TZNF is more, maybe a bit more fluid-like. And it's also showing in the rheological the results. And rheology, it's a study of flow and deformation of materials. So here you see three variables: critical stress, the storage modulus, and the loss modulus. The critical stress is now the blue object. The storage modulus, or the solid-like component, is the black object, and the loss modulus, or the liquid-like component, is the red object. And there is two things I want to show you here. First one being the blue objects. in the left column in TZNF, they're higher than in the right column in the alginate. Now this means that the TZNF matrix under shear stress like this, it doesn't break as easy as the alginate matrix. It's really nice because in photobioreactors where we have the mixing, the matrices will need to survive some shear stresses. And the second thing is that the black objects are in the rightmost column, they're higher than in the TZNF column. And now this means that the calcium alginate matrix, it is very rigid, but it's also brittle because it breaks down easy. And now look at this, it's beautiful. The blue line, which being the TZNF matrix, it's always above the reference material. The alginate matrix, specifically in these conditions, it actually breaks down. So we have there bicarbonate in the ethylene producing system, which chelates the calcium ions from the hydrogel and it breaks down the matrix. So this TZNF matrix really is stable through multiple production cycles. So we can produce ethylene for prolonged periods of time. All right, so you've seen now alternative way to sustainable bioproduction and energy production with these photosynthetic cell factories. We really try to overcome bottlenecks by moving from suspension to solid state inside these systems. And we want to use nanocelluloses and cellulosic materials like CNF here to mimic the leaf architecture. And already we've seen that, for example, these TZNF-based hydrogels, they really offer well-performing alternatives for these existing materials from alginate by mechanical point of view and also from the production point of view. And in the future, of course, via synthetic biology, we can engineer the cells even more for different kind of products. We can modify the matrices even more to get them to be more porous, more mechanically stable even if we need it, or something completely different. So it's a beautiful combination of material science and biotechnology. I'd like to thank our funding organizations and all the collaborators, and with this I would like to thank you as well, and if you have any questions, please you can ask me now.